96% of medical students at UCLA did some sort of research. That number goes to 98% at Northwestern and 99% at Harvard. While this doesn't mean that research is required, it surely suggests that getting into a lab can help you become a doctor. So you frantically open your email and get back to that pineapple photosynthesis lab that you saw in today's email newsletter, which, by the way, also went to 1,000 other premeds. But you're determined, so you look up 10 research faculty and send each of them a cold email. 3 weeks pass and nothing. 8 of them don't respond and 2 of them say they just don't have space right now. Spoiler alert, it's probably because your emails sucked. And that's okay, because I'll show you the exact steps to take today to earn a research position in 3 weeks, even if you have no experience. And this video comes with the exact template I have used to help dozens of pre-med students earn research opportunities over the last 4 years. Let's get started. All right, so I figured what better way to show you how to get research step by step than just literally doing it alongside you. And so I have opened a completely blank Excel sheet here, and we will start from the top on just how simple it is to get a list of researchers together and how to reach out to them. So step one, you're gonna have to create a list of opportunities, whether it's professors or research PIs or faculty members or clubs or minors. I think this is a good place to talk about where can we ever find research opportunities. And I think the big buckets, at least, are clubs. Clubs, like I'll use UCLA as an example, but there are protected clubs that already have research opportunities. For example, CTSI Research Associates, EMRA, Student Stroke Team, Student Stroke Force. Clubs like this, where you can see on the website here, they're formal programs that pair you with a PI or pair you with a project that you can work on. So that's an option always. It will vary certainly by campus, and if your campus has 0 opportunities, which I highly doubt, but 0, even if it's 0, you can still find other options. So that's clubs. You can also take a look at dedicated minors or programs or research internships. I think at UCLA there is a biomedical research minor, which I was a part of. But there's also programs like BICEP at UCLA. I forget what it stands for. Let's see. I just remember that it was a very interesting name. Biomedical Sciences Enrichment Program, BICEP, which will pair you with this, it will equip you with the skills to be involved with research. Okay, so you can look at formal programs like minors and summer programs and things of that sort, and then you can look at clubs that have protected research. Next, you can look at a campus's undergraduate research center or portal, and we'll do that together. And then we can also ask upperclassmen or juniors or seniors who are graduating soon and would know if their lab has any openings in the near future. Okay. And lastly, probably the highest yield one is just Google, right? Departmental websites with faculty directories. And if you don't know what to Google, there's kind of an easy framework that I like to use, which is you can start with your major, because all majors will have an associated department or faculty of professors. You can start thinking about people, populations, or problems. I call them 3 Ps, which is just a nice way to think about some of the things you might be interested in researching, and then just overall, very generally, your interests. So we'll go through all these examples just to show you how I would develop a list. And you can notice I'm no formatting here, no fancy code, no colors, just writing things down and seeing what sticks. So let's move on here and do a little bit of Googling. So I showed you the specific the formal programs that you could get involved with. That's there. I Googled the UCLA Research Minor. You can add any campus that you are a part of, and you'll see the UCLA Biomedical Research Minor here. And that has opportunities once you get admitted to join labs on campus. In fact, I read a little bit more about it, and they pair you with a research mentor that would best fit your interests. Okay, so there's minors like that at UCLA neuroscience major, that's what I was, and I just added literally the word research here. So you can kind of scroll down. This is like required research, fine. And this is like the department requirements, major capstone, and then you start getting things like this. Neurology and neuroscience research, or UCLA neuroscience research page. Maybe click some of these things and you'll see, oh, interesting. The medical department, like the neurologists and neurosurgeons, have research ongoing. And you can see on the left here, there's a bunch of different research innovation categories, from brain mapping to epilepsy to movement disorders to neuroinfectious diseases, neuromuscular diseases to stroke. We already see 4 labs here, including Dr. Jason Hinman, who read one of my undergraduate theses. Resident research projects. Residents are a great person or people to get connected with because they have the research projects. They just don't have the time to complete them. And you have the time to complete them, but you don't have the research projects. And so just going through that, you can open up faculty, you can open up researchers, fellows, research trainees, open all those guys up. You can even open the Hinman Lab, see if you're interested. And so you can see I'm pretty flippant about this. I'm scrolling around, seeing what I like, seeing what I don't like. Jason Hinman, he identifies new molecular pathways that link stroke with Alzheimer's disease. You can read a little bit more about his research. This is his lab beach day, so you can imagine he has a pretty big lab. Probably a fair amount of undergrads or graduate students. Here's Brian Toe. He looks a little bit on the younger side, so maybe he's a former undergrad in the campus. They went on an escape room together, et cetera, et cetera, et cetera, et cetera. So postdoc fellow, this would be someone that you could reach out to, right? If Dr. Hinman doesn't respond. Brian Toe is this undergraduate student that you could reach out to as well. All right, so then, I mean, just to be thorough, I would add, I forget the guy's name, but postdoc, Jason Hinman, Brian Toe, just things like this. I'm just starting to write down things as I'm scouring. Looks like I opened a research tab. I can scroll down. I'm not even reading these things, right? Like I'm just browsing right now. These are probably the chairs of the department, how to get into research, okay, requirements, research resources. Oh, there's an undergraduate research center. We'll open that up. PhD faculty list, it looks like I've already opened that up in the past. And then Brain Institute members. So these are probably just faculty members from different departments across UCLA that do research. So members by research area. Ooh, now we're really starting to see things, right? Maybe I'm interested in addiction or learning a memory, or maybe I'm interested in synapses circuit or neurotechnology. Let's say in high school I was big on computer science, and now I'm interested in neurotechnology. Click on that neurotechnology guy and see what we get here. So we get Asaf Bari, Ian Cook, Benjamin Arani. And so now we're starting to add these people to our list because these are all people that work in neurotechnology. You don't even know what this person works on. Unfortunately, just a picture and a title here. But that's okay because all we need to do is just look the guy up, right? So let's throw him on our list as someone to research in the near future. And then maybe you continue to get 50, 100 people, and now you're working your way through it. And let's just get a rough sense of what he does. Research lies in the intersection of engineering, neuroscience, and physics, applying tool development methodologies. Let's see. Develop a Miniscope, an open source microscopy platform for recording and analyzing neural feed activity in freely behaving animals. So he looks like he makes technology or these microscope platforms so that people can better understand neural activity in mice or animals in a way where they're not like tethered to a machine, or they don't have like an open skull that you need a microscope for. And so I'm just going to keep that there. Okay, great. Now we're continuing to go down. Undergraduate research portal. All we really need to do here is just log in or getting started, and then you'll see all the different research programs that are available. UCLA faculty, I love this part here because it shows you the new members as of December 22. And guess what all new members need? New members or new PhD professors will need manpower to run their lab. So Tiffany Ho is new here. She got her PhD at UCSD. And she has the Candy Lab, Cognition, Affect, and Neurodevelopment in Youth. So she works with youth, adolescent development, stress, depression, big data. Read a little bit about her biography. UCSF, okay, and then you can open up her CV. You notice, again, I'm browsing. I'm looking for things that'll catch my eye. So, Tiffany Chang Ho from UCSD, trained at Stanford, was from Berkeley, cognitive neuroscience, a lot of National Institute of Mental Health, a lot of grants here. Let's see what work she recently did. So just scrolling. Roles in inflammatory and glutamatergic processes and neurodevelopment underlying adolescent depression, right? And she got $667,000 for that 5-year grant as the PI. So I forget her last name. I think Tiffany Chang Ho. We'll just stick that there, right? Just for us to remember. And we're continuing on research development track. Okay, so we have a couple of R25 K grants. Some of them were residents, so these are more people to reach out to, right? These are all people who now have labs. Dr. Ziegler is actually my former mentor. And then these are people who are current fellows, so they're currently working on their research. I've worked with Dr. McCrinnan in the past on my neurology rotation, and we're just going forward. Okay. So now we go to the Westwood Neurology faculty, and again, we see more people. I've seen Amit Arak probably 3 or 4 times now. Gal Baton has taught a couple of lectures at UCLA. So even if you don't write everyone's name down, right, just the sheer volume of people here, there's just no way where you cannot find someone that you're interested in working with. And this is just neuroscience and neurology. There are so many different other fields of interest at UCLA. So I'm just adding people, and you can Google these people later. And research academic series, more people, right? So these are these 8 or 10 people on the research track, all PhDs. Click on one of them, for example, Yuichiro Ito, who does multiple sclerosis work. You just scroll through some of his publications. Parent of origin, T lymphocytes, okay, CD4 plus T lymphocytes, autoimmunity, sex difference, oligodendrocytes, neuroprotection in multiple sclerosis. So a lot of these, honestly, for me, is kind of too complex. I don't really understand what his work is, so that's okay. I'll just keep on pushing. This is the page we saw earlier. This is the page we saw earlier about how the different departments are broken up, right? Maybe I'm also interested in computational and systems neuroscience, and I would do the exact same thing where I'm moving through the researchers. And this is a list of faculty from Dean Buonamano, who wrote a book and is a fantastic professor that I would click on and look, and you get his profile. And so maybe I add Dean Buonamano here, and you start learning a little bit more about him. He has very interesting research on time and temporal processing, the ability to tell time. And you don't even need to summarize these things, right? You can just copy and paste some of the most interesting stuff for now, just so you remember roughly what it is that they talk about, and then you can decide if you're interested in them later. Talked about this faculty page already. All right, so that is just an example of how I would personally navigate my departmental website. And we just started with major, right? That's just this one. Now we can go through people, population, and problems to give you an example of how you would use that framework to find even more folks to reach out to. Let's move on to using people, problems, and populations as our next useful framework to finding even more people and faculty members to do research and find research labs to partake in. So let's say I'm interested in, infectious disease because I read a book in high school that really opened my eyes as to how infectious diseases operated globally. So I might Google UCLA research, infectious disease. Click that guy, get a list of faculty members, or really just the infectious disease homepage. You can see visit our physician and faculty pages to find links to their recent publications that popped out. And just click on this guy right here. Paul Adamson, who is interested in sexually transmitted diseases, treatment and prevention of HIV. He went to UCSF and trained at Yale, and then did ID fellowship at UCLA and UC Berkeley, undergrad and master's. He's from Northern California. We can take a look, keep scrolling down. I think there's a button somewhere here to talk about his research. Yep, research there and areas of focus. So you can click on his bibliography here and see, oh, okay, so he's interested in machine learning to predict self-traction resistance in Neisseria gonorrhea, discovery of a novel sublineage multi-drug resistant Shigella. So some of these things are a little bit too complicated for me, so I'm just going to scroll past them. And then screening for monkeypox viruses in men who have sex with men in a sexual health clinic in Hanoi, Vietnam. Okay, so I mean, I understand that's a pretty simple title, right? So I'll read this. This is in Lancet Microbe. Sounds like a very, very good journal for infectious disease. And unfortunately, there's not really there's not an abstract or anything like that, but maybe I can click one of these guys because it's open access. And you'll see I'm just really browsing around. So Paul Adamson has this research that he's working on here in the Lancet. I'm going to stick that link in here, and then I'm just going to read a little bit about this paper. So in 2002, monkeypox cases occurred, 81,000, disproportionately affect gay, bisexual, and other men who have sex with men. Asymptomatic monkey virus infections have been reported. No reported cases of monkeypox in Vietnam, and little diagnostic test capacity. So this study was to establish if undiagnosed and asymptomatic monkeypox infections occurred in these populations enrolled in an HIV PrEP program in Hanoi. So it looks like they went to this clinic and see the prevalence of monkeypox here, given that in Vietnam there wasn't a lot of testing available. So I kind of summarize that there, throw that there. 152 people, about 25.9 years age. And it looks like median number of male sex partners was 1 and 15 reported having group sex. Previous 3 months on the last day of specimen collection. We have 24 reported rectal symptoms. 32 were positive for C. trachomatis and 23 for Neisseria gonorrhea. And then the rest were negative for monkeypox virus. So they were not detected in rectal specimens from these 152 folks. So it's pretty interesting, right? We're giving the diagnostic capacity to this clinic in Vietnam, and it looks like in Vietnam that 150 sample were all negative for monkeypox. So just continue to move along, right? So if we go through infectious disease as kind of our specialty of interest, we can think about if we're interested in LGBTQ health, that would be a good, again, kind of a similar Google. Let's say we're interested, we'll move down now from that framework just to pure interests. Like I personally am interested in sports medicine and sports physiology. So just Google that, UCLA Sports Science Research, and we have the Sports Performance Lab, UC Fit Exercise Lab, UC Fit Digital Health. So I just clicked on a couple of these things. And again, you'll get a sense of, even if it's not direct wet lab clinical research working with patients, there are clubs on campus that do scholarly work that honestly would be better for you just because they're aligned with your interests. From ideation to research and program design, the labs discipline to bridge the gap between lab research and real-world application of health fitness practices. This is essentially a digital health lab, which would be very interesting. So the fitness professor, Dr. Brett Dolezal, and then he has a bunch of these programs. You can get a sense of what they're working on. This is a demo of a VO2 max, where someone is sprinting at the top of his lungs, and the data is being plotted. It looks like they're probably measuring some CO2 out of that machine there. This is a couple of the interns. There's a fourth-year HBS major who likes to go bowling and wants to be a physician assistant specializing in anesthesiology. Me too. And then this is Dr. Dolezal, who runs the lab. Very, very interesting. So this is measurements of, that's that new sports equipment that's tonal, I think, that LeBron was using. So again, we can throw a couple of these things here, put that there, take that down. Sports Performance Lab powered by Exos, a team of coaches to help you and your team get ready for the moments that matter. So you get a sense, looks like it's more like UCLA health and professional sports teams. And so this is, interesting. It's a sports facility that's located at the home of Los Angeles Lakers in El Segundo. So it actually would be very interesting if you could get connected and work in a lab.
Home of the LA Lakers, experts in the world, and we have a methodology that's
professional athletes as well as
and even physical therapy. So
we want to help you reach maximum.
If you could demonstrate your interest and they had an opportunity or role or space for you, and so don't count yourself out. I would throw that in there as well. UC Fit Exercise Physiology Lab. Similarly, they have all these health fit assessments. Please visit this site, and so that you can continue to play with these things as you see fit. Okay, so now you have all these research articles, you have all these websites, you have all these professors that do all this interesting research. Now what do, right? The next step is to filter all of these based on your interests, and let's go ahead and do that now. Okay, so we're going to go ahead and filter these now. You'll recognize that when we started with basically every option to get research. And that included clubs, that included minors, that included programs, that included the undergraduate research portal. And then we ended up doing a little bit more of a focused search based on our major, and then eventually our interests. Let's say we're majoring in neuroscience, but we might be interested in infectious disease, or we might be interested in sports science a little bit more than we are in neuroscience. And that's totally okay. That's very normal for people to have interests outside of their major. You'll notice that when we go ahead and go through the line, some things will be more interesting than others. So let's say, for example, I'm extremely interested in neurotechnology as opposed to emergency medicine, for example, which is EMRA. Put that here. And even though I'm a neuroscience major, I might be less interested in stroke and more interested in neurotechnology. We don't have to do a lot of filtering here, but let's say we had a couple of these people. We'll kind of highlight these 6 neuroscientists and his postdoc as well. So that gives us kind of 7 of our tier 1s, and we'll just make it green. Again, I'm trying to just go through this pretty quickly because you don't need the perfect looking sheet. We're really optimizing for the most important thing, doing the thing here, which is the reach out, the actual email. And so the sheet is a little bit less important to that. You'll also learn that when we get all of these options for research, let's say we also had a couple options that came from our email listserv. So every week the counselor sends out a bunch of opportunities, and let's say in this last week we had like a plant photosynthesis lab, and then we had a zebrafish cardiology lab, and then we had a pediatric stem cell lab. You'll see that these email listservs are they're one size fit all, right? This opportunity is not specific in any way to our interest. It's just literally put into a blast with thousands of premeds, and the lab is looking for kind of just a warm body. And so this is why these things shouldn't make your tier 1. And it's probably why when you tried to apply to these labs, you didn't get in, because your resume is not aligned, your interests aren't aligned, and you probably didn't respond the first or fastest out of all the people who saw the email, out of the 1,000 undergraduates at UCLA who saw the email and immediately responded back. And that's a good thing, because imagine if you were to be the one who got that position. Now you're a guy who's interested in neurotechnology and sports science, but ended up at a zebrafish cardiology lab. You spend 2 or 3 years there and you're not as fascinated, you're not really interested, so you put in the bare minimum work and you get the job done. But when it comes time to talk about it during your personal statement or your secondaries and your interviews, you didn't like it at all, and that is apparent. And that overall reflects really poorly on you, because if you spent a little bit of extra time up front 3 years ago, you could have been in a lab that really excited you. You could have been in a place where you felt like you were supported and the mentorship there was helping you develop as a researcher, and your projects were better. and you had more fun and you spent less time, right? Those things are the things that stand out over time, especially when it comes to medical school admissions. The same goes for these clubs. While these clubs can be quite impressive, if you're not interested in translational research or emergency medicine or stroke, then it is to your detriment to join these clubs, right? Even if they promise a research opportunity, that research opportunity is not a good fit for you. Make sure that you prioritize your actual interests and you think a little bit, a couple of minutes here, just trying to figure out what excites you about in class, what topics you think roughly after reading one article is like, whoa, that's kind of interesting. I could see myself studying that a little bit more. That is how one of our application breakdowns, also a Bruin from UCLA, ended up at a fantastic research opportunity. I'll just show you a diagram that I drew about him here. But there's publicly available research kind of on the email listservs and on the the undergraduate research portal and from upperclassmen. You get these random ones like aquatic crop cultivation, skin cancer immunotherapy, epigenetics and stem cells, RNA virus breakdowns. And these are all the pre-meds on campus that also are interested in these publicly available options because they're on the portal, they're on the email listserv. It's easy to just click apply or send a quick email and a resume. And so everyone applies to these for. This student, we'll just call him Jack today. So Jack recognized that he wasn't interested in these things. He was a football player in high school and so was really interested in sports. A lot of his volunteering experience was with the homeless population. He himself saw one of his best friends in high school who had NFL dreams get too many concussions and have to end his career. He was an EMT himself, so a lot of acute care, and then he worked a lot with children. So he was thinking like, these interests don't really align with these things, and so he wasn't going to play that game and apply to those positions. Instead, he looked for something that was within his interests of football and concussions, and he found this interesting equipment manager position where they install accelerometer sensors to measure head impact. So it was essentially basic science or imaging research for athletes who had received a concussion. He facilitated MRIs and head ultrasounds to track brain activation over time. This research position was not publicly available, but it is, as in, you could find it on Google, as in people were doing this research, but it wasn't a lab saying, please come, we need a warm body to help us. He reached out to the PI and the postdocs and the graduate students and got involved with that research. And it ended up being one of his most significant activities over the 2, 3, 4 years he was at UCLA. This is a great example of taking your interests first and starting there first and then finding research positions that match that, as opposed to what most pre-meds do, which is the opposite. They start with whatever is available, regardless of what their interests are, and then they apply broadly and try to get into one of these positions. And when they don't, because they're not a good fit for those positions, they ask, why can't I get research? Well, it's because you're not very, very interested in these things, and that comes across in your application. And even if you got the position, in 2 or 3 years down the line, you will pay for having a poor kind of interest position mismatch. Okay, so I'll take that and go back to our simple spreadsheet that we've been working on here. And recognize that once you find a good fit, when you have that lab like Jack did that's really in alignment with his interests, or when this student, we'll call her, we'll call her Benita. Benita's resume, which I have reviewed in another video, you can see here, this is probably on the order of a dozen, maybe 10 publications and presentations. And you'll see that this is what world class looks like. This is impossible for anyone who's doing research in something that they don't like, right? This is only possible if you're invested and interested in what the lab does, in the questions that the lab is solving, because then the lab members want to invest in you. They want to develop you as a researcher, and you want to kind of meet their interest in you with your own interest. And you get this positive feedback cycle of you get more time in the lab, you get more attention in the lab, so you get more training in the lab. And all of those skill development over years leads to all this productivity. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different publications and presentations. And you can imagine why a top 5 Ivy League medical school wanted this student. Breast cancer, woman with early stage hormone positive breast cancer, metastatic breast cancer, chemotherapy resistance in breast cancer, fasting in breast cancer survivors, some receptors in multiple myeloma. So a ton of cancer-based research. And this, again, only happens when you have the right fit. Okay, so that is why tiering or putting your opportunities in priorities is important, because you can't reach out to all of these people equally, but you should reach out to the ones that are most interesting to you up front and with the highest quality. So that is step 2. Now we'll talk about step 3, how to earn the research position. Essentially, this boils down to how do you demonstrate your interest in a high quality outreach email. And before we get there, let's just kind of flip our perspective for a little bit. If you are a professor or a PI or a head of a lab, you would want someone who is resourceful, hardworking, and proactive, right? And how do you demonstrate that? Well, you can demonstrate that by, first of all, being a person who sends a cold email that is well researched, sending an email when the lab didn't ask for anyone at all, right? A lab would expect emails from undergrads if they put a public posting looking for work, looking for support online. But if you're a lab who's just really operating day to day, doing their projects, and then an interested undergrad reaches out to you, then that's unexpected because someone found you, did research on you, and took the time and attention to really draft a strong email. So I find that a lot of pre-meds get stuck on how to do research. So we will do that together. Let me see. I essentially just looked up one of my own old papers in a lab that I was a part of here. And what I will walk you through is how to read an abstract just really at the level of what we need to. It essentially gets the main conclusion of the paper and then express to the professor in an email that we read it, that we were digesting it, and that we're interested in the work. So let's read this paper together. It is titled In Vivo, which let's say I'm a freshman and I don't really know what that means. That's okay. Mouse, I know what a mouse is. Mouse model of spinal or spine implant. I'm not really sure what an implant is. And then infection. I do know what infection is. Okay. So spine implant infections portend poor outcomes as diagnosis is challenging and surgical eradication is at odds with mechanical spine stability. Okay, even that I don't fully understand. For example, I don't know what the word portend means. Let me just look that word up. Portend is a sign or warning that something bad is about to happen. Okay, now I get it. So if you have an infection of an implant, still don't know what an implant is, but maybe we'll figure that out or look it up later. If you have an infection of it, then it sounds like that's not good, and surgical eradication is at odds with mechanical stability. So it sounds like if you do surgery, that is a problem because you can affect the stability of the spine. I don't know why that's a problem, but it sounds bad, right? I'm just kind of operating under the assumption that I don't know a lot here, and you won't, right? You're a freshman who just took general chemistry. Reading a sentence like that is kind of out of your current scope, and that's totally okay. I'm reading it through to show you what I would be thinking. The purpose of this method is to describe a novel mouse model of spinal implant infection, or SII, that was created to provide an inexpensive, rapid, and accurate in vivo tool to test therapeutics and treatment strategies for spinal implant infections. Okay, I kind of understand that. Let's see, first of all, what does in vivo mean? In vivo means in a living organism as opposed to what? So if I look, what is in vitro and in vivo? In vivo means in a living organism. I'm guessing this is the mouse. And in vitro means in a dish or test tube. Okay. So this is a little bit maybe more realistic because it's not on a test tube, but it's really in a living thing like a mouse. And this is a model of infections in a live mouse that is cheap, fast, and accurate. Okay. In this model, we present a model of posterior approach. So from the back, spinal surgery in which a stainless steel K-wire is transfixed into the L4 spinous process of a 12-week-old something mouse and inoculated with or injected with something CFUs of a bioluminescent strain of Staphylococcus aureus Zen 36 bacteria. Notice that I'm not really that interested in the really complicated jargony stuff like C57BL6J. I don't need to really know what that is, or a CFU, or a Zen 36 bacteria. I'm just trying to get a big picture of what this is. So like, what is an L4 spinous process? So an L4 spinous process is essentially, it's one of the bones of the spine. So they're just saying where they are injecting the bacteria. Okay. Mice are then imaged for bioluminescence. So I know bio means life, lum means light. So I kind of don't really understand it, but that's okay. Let's move on. But I'm moving quickly, right? I don't need to stay so hung up on everything. My goal is not to learn everything, but to get a broad picture. On post-operative days 0 through 35, imaging signals from a standardized field of view are quantified to measure in vivo bacterial burden. So this imaging is supposed to tell me something about how much bacteria there are. Okay, I'm kind of getting it because bacteria are related to infection. To quantify bacteria adhering or sticking to implants and peri-implant, or peri meaning around implant tissue, mice are euthanized and the implant and surrounding tissue are harvested. So the mice are killed and then the implant in them are taken out. Bacteria are detached from the implant by sonication. Okay, that sounds kind of important. So what does that mean? Sonic means sound energy to agitate particles. So they probably just like vibrate them to get all the bacteria out. Cultured overnight and then coliform. Okay. The results acquired from this method include longitudinal bacterial counts as measured by in vivo Staph aureus bioluminescence and CFU counts. Okay, I don't really understand that. That's okay. While prior animal models of instrumented spine infection had involved invasive ex vivo tissue analysis, the mouse model of this in this paper leverages non-invasive, real-time, in vivo optical imaging of bioluminescent bacteria to replace static tissue study. Okay, so this sounds like kind of the conclusion, right? So if I can understand this sentence, then I think I'll go pretty far. While other animal models of spine infection have involved invasive, ex vivo, if in vivo means in a live organism, let's see what ex vivo means. Ex vivo means out of the living, where they probably had to kill the mouse early and then take out the stuff early. The mouse model that we have leverages non-invasive, so you don't need to kill the mouse, you don't need to take anything out. Real-time in vivo optical imaging of bioluminescent bacteria to replace static tissue study. Okay, so it sounds like this is more beneficial because you can see the bacteria at different days without having to kill the mouse until like the very end. Applications of the model are broad and may include utilizing other bacterial strains, incorporating other types of genetically engineered mouse to study the host immune response, and evaluating current or investigating new diagnostic and therapeutic modalities such as antibiotics or implant coatings. Okay, so I kind of have a rough sense. This is probably an orthopedic spine lab that works on infections in the spine. And if we need a little bit more support, one thing that I am a very big fan of is just summarizing this in a 5th grade level. And we have technology nowadays that can help us with that. This is a large language learning model that you guys are certainly familiar with called ChatGPT. I created this yesterday, mouse spine infection study. And you'll see I just wrote this prompt. Please explain this research abstract at a 5th grade level, and then I just copy and pasted it. So I think this is kind of really funny what it says. Let me just blow it up for you so you see it. Okay. So it says, all right, buddy, let me break down that scientific jargon into something simpler than shooting hoops. I think it's understanding that I'm a big fan of basketball. Imagine we're talking about a game of hide and seek inside a mouse's body. Scientists want to figure out how to deal with infections around these tiny metal things they put in a mouse's back. I'm guessing those are the implants. These metal things are like superhero backbones, but can sometimes get infected. Detecting these infections is tough, and fixing them with surgery can mess up the mouse's spine stability. That's kind of what we got in the beginning, right? Now these brainy researchers came up with a cool way to test medicines for these infections without hurting the mouse. They did a mini surgery on the mouse's back. They put a small metal thing in there and infected it with glowing bacteria. That's the bioluminescence. They then used special cameras to watch the glow inside the mouse, kind of like seeing the glow in the dark during hide and seek. Interesting. this helped them see how bad the infection was over a few weeks. So you can see how the infection grows on day 0, 1, 3, 7, 9, etc., etc. So after a while, they took out the mental thing, they counted the bacteria and checked if the medicine worked. This method is like a superhero tool to test new medicines for these spine infections without causing too much trouble to the mouse. So in simple words, they created a game of hide and seek inside a mouse to figure out the best way to fight bad infections around its spine superhero metal thingy. Cool. All right. So it's not totally accurate, but I think it gets you, and I know it's not totally accurate because I worked on this project and it's not quite a game of hide and seek, but it is very good at getting you pretty much 80% of the way there and understanding why this model is important and how we use it for different things. And so that is a way to easily and simply break down something that's not a lot of text, right? Probably one paragraph, but very complicated, especially if you're not in the field and haven't studied it, into a way where you can roughly understand. And so now if we were writing a email for this lab, and let's say this is, I don't know, Jason Hinman's lab, I would just write down some questions that I had after reading the summary from ChatGPT and then reading just kind of the abstract on my own. So some questions that I had, because they talked about antibiotics and implant coatings in the end, I'd be like, what antibiotics or implant coatings are you currently testing? Stick that there. And then my other thoughts were like, oh, you guys are testing some antibiotics and implant coatings. That's probably because humans use them. So my next question was, are any of these antibiotics or implant coatings available in humans? And then the last thing I was wondering was, these are spinal implants, but don't humans get other types of implants too, like knees and shoulders? And so my other question was, do other implants have similar problems, like knee and shoulder replacements? Again, just top of mind, the first couple of questions that come to mind. These are not the most specific, exciting questions for a research PI. However, they do a good job of demonstrating that you have read the paper, that you demonstrated the interest, and you took the time to try and understand it. All right, so now that we've done our research, we have a set of questions and a nice little summary to work with. Let's write an email and contact the person. Right? In this case, let's just assume that we're reaching out to Jason Hinman here. You can just look up Jason Hinman, email UCLA, and see what we get. If we click on this first one here, let's see what we got. He has a website. Let's see if we can find an email pretty quickly. Their email, not on this one. So now we'll look down here to see on his personal lab website, see if there's a way to find a nice contact information. Again, I'm showing you from beginning to end how I would be going about reaching out to these people. He has a Twitter, so a Twitter DM can go a long way as well. Let's see, let's make this a little bit smaller. It's a nice looking website too. Coming down, a lot of people in the lab, right? This is a good sign because it demonstrates that they have, Dr. Hinman has experience kind of working through undergrads and supporting undergrads, reaching out to the lab. So there's some UCLA undergrads interested in volunteering in the lab. There's a Google form here, and there's his email, hinman.uclalab@gmail.com, and there's stuff like this. Okay, so you can certainly reach out through this form. You can reach out through this Google form that he's already created. You can reach out through the email. All those are possible options. You might do all 2 or 3 of them, depending on who you are reaching out to. So I want to show the spectrum of, look at this, are you interested in other labs? 10 hours per week, great, fantastic. So now we will take a look at how to write an email. And one way that I think would be a good educational experience is just demonstrate some of the other emails that I've written in my past and show you why they were good and not good, and then show other emails that I've seen to demonstrate what you should be kind of emulating on your time. So I'll start with this one end of the spectrum where it's a little bit too little effort, and then I'll show you what too much effort looks like, and then we'll end kind of in the middle with a nice moderate email that works very well. Okay, so we'll start with, on this end of the spectrum, an email that probably showed very minimal effort, very generic. It's almost like they copy and pasted a template from Reddit or ChatGPT or something like this. We'll read it together here. Hey, Dr. Lee, my name's Harold. I'd like to volunteer in your lab. I major in neuroscience. I've completed Gen Chem, Bio, and OChem labs. I'm looking to spend about 5 to 10 hours per week over the next 3 semesters as I prepare to apply to medical school. While there are many other premeds trying to get research hours, none of them offer my drive for prostate cancer research. I'm interested in your research on the life-changing effects that prostate cancer research has on real patients. The possible applications for persons diagnosed early with prostate cancer are so exciting. I'd love to be a part of the team. I'm ready to learn from you in your lab, and I hope to hear back soon. Sincerely, Harold. Okay, so off the rip, the thing that I notice is that this email is all about Harold, right? I would like to volunteer. I am majoring in neuroscience. I have completed. I am looking to spend 5 to 10 hours. I'm interested. I am ready to learn. So everything is about what Harold wants, who Harold is. And if I were a professor or a head of a lab, one thing that's important is that you sent me an email asking for my time, my expertise. I've been in this field for 10, 20 years, and I have a working lab, like currently without you, right? And so my approach, and some other folks may disagree with this, but my approach is to focus on them, right? Their interests, what they need. If you have certain questions that are well educated, well informed, throw those in there. But the majority of the email, especially not what you start with, it shouldn't really be about you, right? Your interest to volunteer in the lab, your career goals in medicine. While I appreciate the transparency, I just think this is an email that feels like you can literally copy and paste it to anyone. All you need to do is change this. Instead of prostate cancer, just change it to whatever research they work on. And you can repurpose it. And while that may sound efficient, it's actually ultimately extremely inefficient because your response rate will be essentially 0 with this email. If I read this, I wouldn't be very interested in meeting Harold, just because it didn't seem like Harold is very genuine. Harold put in a lot of effort to get to know my lab. And so I'm going to now show you the other end of the spectrum where I think I did too much. And this is actually a real email that I sent in the past. So a lot here. Let me zoom in so you guys can see it. Okay, here we go. Subject line, potential undergrad research opportunities, Michael Lee. My name is Michael Lee. I'm a first-year undergrad pursuing a degree in neuroscience here at UCLA. I was first introduced to your lab through my mentor, Tyler Landman, who is one of your former research assistants. I have enormous interest in neuroscience, and I hope to join your research team. Incredibly interested in 3 of your lab's projects: molecular memory systems in neurorepair, specifically the tonic GABA sibling and AMPA receptor sibling, post-stroke neurogenesis, and developing stem cell therapies that can flourish in the hostile environment of the stroke cavity. The Carmichael Lab research on molecular memory systems fascinates me because of the immediate applicability. Traditional medicine has indicated that typical CNS injuries recover best after being exposed to movement patterns. And the lab builds on this knowledge by looking for a pharmaceutical way to enhance these inherent processes. I'm fascinated by cellular signaling systems, timing component, antagonistic pleiotropy. Oh my gosh. Early GABA and AMPA effects are beneficial, but prolonged effects are highly detrimental to optimal recovery. Just a lot of words here, right? Furthermore, lab's research on neurogenesis after stroke piques my interest because the knowledge of the signaling systems responsible for this process will inevitably open the world of stroke recovery, elucidating endothelial cells and neuroblasts, interventions, post-stroke neurogenesis. Oh my gosh, keyword after keyword after keyword. Stem cell therapies, human-animal translational obstacle, I don't even know what that means, and I wrote it. Valid modality for stroke therapy, a translational bottleneck to stroke therapy. And after perusing your lab's website, talking with Tyler, I would be a great fit for your lab in terms of culture and scientific interest. I am dedicated, I am organized, and I am most importantly genuinely curious about how the brain functions. I've attached my CV. Let me know if there's any way I could join the lab. So you can see, this is a lot, right? Not only did I read 1 project, I read 3 projects, and I wrote 3 paragraphs on each of these projects. And to be honest, even though I appreciate the level of effort and attention and expertise, and not expertise, but attention and effort that I put into this email, it's a lot to read as someone who's busy running a lab. right? You could get away with a lot less, and not only would it be easier to read from Dr. Carmichael's perspective, but also just probably more efficient on your end, especially if you're going to send 10, 15, 20, 25 different of these emails, right? This probably took me 3 or 4 hours to read, write, and that was prior to ChatGPT, where I was trying to study and understand everything. I have no idea what antagonistic pleiotropy was, but I did back then. The other really good thing that I did here was I had a connection through Tyler Landman, right? And if you remember in the simple Excel sheet that we did together, you saw Jason Hinman, but also Brian Toe was his lab, which is an undergrad. They had a postdoc in his lab, and then we looked at his website together. And look at all these people in his lab. So you may know Roy, you may know Jennifer, you may know Davis, you may know Eleni or Christina. All these people, or Manu, who's an undergrad, all these people are different options, especially if you have talked about their lab experience with them. You can get a sense of how Jason Hinman, Dr. Hinman, runs his lab, and use some of those things as entry points into the email or points of discussion. So again, a good thing that I did, connected with a well-known name. I wish I put that name in the subject line, because remember, the subject line is what most professors will read only if they ever open your email. They at least read the subject line. So making sure that you have the most important information there is going to be helpful for you. Notice that my name is probably not important to them, and certainly this isn't important. Potential undergrad research opportunities, that they can get to if they ever open my email. But if they don't open my email, this is actually never important to actually read. So if I did this now today, I might change the subject line to referral from Dr. Tyler Landman. And that would be like a very curious subject line that I imagine most people who worked with Tyler Landman would want to click on. Okay, so that's an email that is kind of on the other extreme end of doing too much. And so now let's kind of read an email together that I think is Goldilocks, straight in the middle, perfect for what we're looking to do here, where it's efficient, but also demonstrates a highly effective email. So let's read this together. Subject line: what is a healthy caregiver relationship? Caregivers for cancer patients. Hey, Dr. Brady, I hope this email finds you well. I was reading your research article from 2005 titled, right from the start, I was surprised to see the statistic that 3 million people care for someone with cancer. That's a lot. 3 million people have cancer or more, and 3 million others are caring for them. I thought that's an interesting insight, right? Not only are there 3 million cancer patients, there's all those 3 million cancer caregivers. I thought the article was very insightful because most people overlook the caregiver. I certainly did, just thinking about that, and their mental and physiological needs. I thought it was interesting how caregivers can experience physiological symptoms like increased risk of cardiovascular disease. I was reading how the goal was to study the relationship between caregiver and cancer patient. Also interested in how it differs based on the age of each person and overall relationship. For example, if the patient is a kid and the caregiver is a mom, and vice versa. Interesting, right? So this paper didn't talk about different ages in caregivers, but this student read it and was wondering, I wonder what it would be like if instead of caring for your mom with cancer, you were caring for your daughter who has cancer. Very interesting change of dynamic. And honestly, the PI, if they read that, they'd be like, that's an interesting study. I might be able to kind of do something like that. So Anna, this student, has already helped and gave value to the professor. And notice all of this is really about the professor's study. It's not about what Anna wants or who Anna is. You don't even know Anna's name in this entire first paragraph, which I appreciate, right? You're focusing on Dr. Brady's work. My name is Anna. I'm a first-year student at UCLA. I was reaching out to not only learn about your research, but also your career. You notice how well that sentence hits? Even if you're very inexperienced, essentially you have no experience, you just graduated from high school, you clearly demonstrate value here. You read the paper, you have an interesting insight, you even offered a possible new experiment or study to do. It doesn't matter if you're a first year or a 12-year-old or a 15-year-old, you're clearly well-versed. I'm very interested in the social work and relationship patterns in healthcare. I think understanding these relations make healthcare more meaningful, more personal. I would love to learn more about your research. But importantly, I would love to hear more about your career. So not only about your research, but just kind of overall your career, where you want to go, how it's progressed to where it is today, clinical research and administrative arms. Okay. I'd love to chat about you. I'm available to meet via Zoom. Okay, great. And here's my availability. Another thing that I love is that they give you availability so that if you're the professor, all you need to do is go, okay, Tuesday at 2, done. Send the invite, all done. You don't have to go back and forth 3 or 4 emails. This is very considerate, right? The student knows that the professor is busy, and so they'll give all the information up front so the professor can decide what to do next. I look forward to hearing from you. Best, Anna. Okay? So that is a fantastic email that I absolutely love. And the last thing I'll highlight here is the subject line. The subject line says, what is a healthy caregiver relationship? Caregivers for cancer patients. And so you'll see that this is just a very compelling question. You want to click on this. It has nothing to do with Anna, it has nothing to do with research, but it's just a question. What is a healthy caregiver relationship? And that would be something that the professor would love to click on because they work on caregiver relationships in cancer patients. And so it's just an interesting segue. Okay, so that is what a strong email looks like, and this is a basic template that I recommend all of our students work through here. Dear research, and you'll see that it kind of mimics what Anna's email looks like because Anna used this template to write a fantastic email that got her multiple research positions within weeks, right? Very simple. The first paragraph is all about them and their work. The second paragraph finally introduces you and why you're interested in them. Third paragraph talks about your availability and how to connect with the PI. And so I will have this entire template that you can use in addition to word for word templates to use for shadowing or phone scripts that you use to call clinics. All of these stuff that I wish I had as a pre-med that are plug and play to get these important clinical research shadowing opportunities, letters of recommendations. All of that will be in one free resource, always free for you guys to use. I'll put that in the link in the description box below so you don't have to type all this yourself or anything like that. It'll be nice and plug and play for you. Okay, great. So let us move back to the other Excel sheet. We're back to the simple spreadsheet. We're working on our emails for these top 7 or 8 folks. And the last thing I want to suggest is remember to use technology to your advantage, right? If you want to simplify your emails, you can always ask ChatGPT to simplify it, make it in a 7th grade or 5th grade level. You can always use Grammarly to make sure that your spelling and your grammar is nice and legible. You can also make sure that you pay attention to your subject line. Remember, when you're looking at an email, let me just put this here, and I will show you kind of what my current email inbox looks like. But when you're looking and you're a working professor or PI, remember that you just get subject lines, right? And so if you look at your own Gmail, all you see is not all the hard work that the student put into the body of the email. All you see are the subject lines. So I'll just show you kind of my subject lines here. Annual planning workshop. Here's your recording. Productivity assessment. 5 life-changing psychology hacks for YouTubers. International, New York Knicks, Equinox, ACA monitor, all this stuff. Upcoming bills, how to allocate your time. If you're a PI, that's all you see. You don't see anything until you click into the email. And so you want to make sure that the subject line is your best work yet. It is so compelling that you actually spend 95% of your time thinking about it because that is what 95% of professors will read. And so I'll put this also in the template for you guys, but here are all the subject line options. You can do some things that will just resonate when you review their website or their research articles. Here are examples that I find compelling, like saw your all Spanish research seminar, or can UCLA students benefit from wellness education? A nice little question. My friend struggles with substance abuse, like if you're interested in studying substance abuse psychology, I'd love to learn more, something like this. CRP and ibutylast, a fascinating paper. You can do referrals, just like I had with Tyler Landman, referral for Tyler Landman. And then this is my least favorite, but this is kind of something simple. This is not as compelling as the other ones up top. I recommend, and I know that you guys can come up with something better than that. Here are some real subject line examples that actually earned real research experiences, so I know that they worked. Single agent immunotherapy, but in rats. Is the label survivor for cancer patients wrong? Like, this is such a very compelling subject line that will force a professor to really be interested in what you have to say inside, because it's such not a provocative, but it's such a well-informed, well-thought question that you want to know more about what the email says, right? How does a child learn to deal with a parent fighting cancer? So please, for the life of me, spend a ton of time with your subject lines, because they may be the only thing that the professor ends up reading. Okay, so I'll leave those for you guys now. And lastly, that's step 3. You send all those emails, try to get all that done maybe within a week, right? You can probably knock out 1 a day or 2 a day or 3 a day, especially if a nice focused template and you do a good job kind of summarizing research articles with the use of ChatGPT or other things. Next, we will go to the most important part after the subject lines, which is following up. Remember that professors are busy. I just showed you my email. I checked it literally 30 minutes ago, and you saw that 6, 7, 8 emails had come in those 30-minute time span. So recognize that professors are much more busy than me. They probably get hundreds of emails every single morning. And so following up is actually not disrespectful. You're not wasting their time. You're actually respecting their time by bringing it back to the top of the inbox to remind them of something that's important to you. That is the mindset shift that really changed how I saw the importance of following up with a professor. So I'll go back to my emails about the Tyler Landman Stroke Recovery Lab, and I will show you how I followed up to him. And eventually it didn't work out, but I do respect the amount of kind of attention to detail that I was giving Dr. Carmichael. I think it's just a good sign if you are on top of it and you're kind of meeting Dr. Carmichael halfway. So you'll see this is my initial email, Dr. Carmichael, in April 2016. He actually responds immediately, and then I respond the day right after, right? He tells me Tyler was a great student in the lab, but he doesn't have any room. Check back in the spring. And look what I did. First of all, I said, thank you for letting me know. I will certainly check back in the spring. And look what happened. I did actually check back in the spring. I guess this is the fall. I did check back in the fall 6 months later. And again, he responds that same day. And again, I thank him for telling me, or he tells me that there's no spots right now, but to check back again in a couple of months. And so I thank him again for giving me the opportunity. It looks like I'm forwarding the email to other people who were looking for like a way to write a good email. I think it's kind of funny now looking at that email, given that it's so long, but clearly it was very effective in getting Dr. Carmichael's attention. And then this is my downfall. I never actually followed up again, right? And it might take 6, 9, 12 months to get into a lab, but this is one of my favorite labs with clearly a PI who had worked with many undergraduate students and many graduate students that have gone on to very successful medical schools. I think Tyler ended up at UCSD Medical School, true to a dream for me. But I stopped following up, which is probably the reason that I never ended up working with Dr. Carmichael. And I wrote here, did this ever get a response? Yeah, it did, but the opportunity didn't work out eventually though. So this is just, again, a great example of the importance of following up. Set reminders. Nowadays, your email providers will have ways to remind you in 3 months to send another follow-up. You can also just schedule an email to be sent in 3 months so that you can write the follow-up now, and then in 3 months, the email provider will automatically send it on your behalf. And if you follow those steps, those 4 steps, which I'll review for you now, which is step 1, find faculty that are options to get research, whether that's clubs, professors, minors, undergraduate research portal. That's step 1. Find them, put them into a simple Excel sheet like this. Step 2, filter them based on your interest. I'm most interested in stroke technology or neurotechnology here. I highlighted these 7. Recognize that emergency medicine is not that interesting to me. Translational research, not that interesting. plant photosynthesis, zebrafish cardiology. Those are all options that I saw, but I'm not as interested in them, and so I won't put them in my first priority. So those are my first priorities. Then step 3, research so that you can write a high quality email. I showed you what too much looks like. I showed you what too little looks like. And I showed you kind of that Goldilocks zone where you're doing a good job of showing your interest, putting them at the forefront, and then respecting their time by giving your availability and things of that sort. I showed you how to do research using ChatGPT, and you saw how quickly I skimmed through things, not really getting bogged down in the details of Zen36 Staphylococcus bioluminescence, but getting a big picture understanding of like, what is this research about? Why is it important? And how I was Googling things that I just didn't really understand. How I used ChatGPT to summarize and get me a better understanding of what was going on. I wrote down some questions for the email that I stuck here in this set of this spreadsheet. Again, it's not organized, it's all cut off, looks a little messy, but that's fine. I copy and paste these questions into the email, or I took a portion of it, and then I showed you a template of a high quality email that just gives really genuine curiosity, and it shows that proactivity that professors are looking for. Most importantly, I showed you the importance of a subject line, and I want to talk about the Starbucks test, which is essentially if a professor saw that subject line at Starbucks while they're waiting in line for their coffee, would they be interested in clicking that to learn a little bit more, right? And most of our subject lines, especially the one that I showed you from my old email, potential undergraduate research opportunities are not very compelling. And so those will not get opened and will not get responses. And then lastly, I showed you the importance of following up and how following up over even a 6 months time got me 2 responses from my #1 lab. And while it didn't work out, how many of you guys are getting true responses, true interested responses from your PIs? I think that the more we can do that, the more consistent, organized we can be about it, the better our results will be. Those 4 steps will, I know, do a good job of opening up research doors so that you guys can get opportunities that you're interested in, and eventually you'll land the one research lab that you're interested in working for, and you'll work there for 2 or 3 years and really develop your expertise, interest, and be mentored to develop into a strong undergraduate researcher. If this helped, the word-for-word research templates, as well as the subject line examples, in addition to a lot of other templates, word-for-word shadowing emails, phone scripts when you're calling clinics. We'll work on adding letter of recommendation emails there for you so you can ask for a letter of recommendation in a very respectful way. All of that will be available in our description box for free forever so that you guys can use and take advantage of some of those things to make your workflow a little bit more efficient, to get you the opportunities so that you can focus on actually growing in the opportunities and building your application. Hopefully this was helpful. This was a super long video, but I think going into how to get research step by step is something that I haven't seen a good job on in the internet. And so hopefully you can see how I was doing my research, how I put things down pretty flippantly, pretty quickly, so I can get to writing that email faster and faster. Thank you for watching, and I'll see you in the next one. Goodbye. Hopefully this video was helpful, and right now you can download that template that I've used with dozens of premeds to get research opportunities for the last 4 years. That will always be free, link in the description box below. Now, if you want to understand just how important research is, I want you to watch this video here, where I review a research-heavy resume that earned multiple top acceptances, including one top 5 Ivy League medical school where they're at now. And if this video was helpful to get research, then this video here will be helpful to get some shadowing opportunities. Thank you for watching this video. Choose your own adventure, and I will see you in that next video.